Hello, friend. Welcome to Science for Sleep. Some people say that a caterpillar turns into liquid inside its chrysalis. It is an image that lingers in the mind, a soft body dissolving completely, becoming something like soup before rising again with wings. That description is not entirely true. A caterpillar does not melt into a formless puddle. And yet, something almost as remarkable does happen. Inside the body of a caterpillar are tiny clusters of cells called imaginal discs. They are so small that you would never see them from the outside. They have been present since the caterpillar first hatched from its egg. Quietly, invisibly, they carry the blueprint for wings, for antennae, for compound eyes, and for slender adult legs. While the caterpillar spends its days moving slowly across leaves, these cells wait. They are not yet building wings. They are simply there, holding the plan. If you enjoy these quiet journeys, you're welcome to subscribe. And if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find their way to this calm corner of the world, one sleepy traveler at a time. A caterpillar is the larval stage of a butterfly or moth. In scientific language, this process is called complete metamorphosis. The life cycle moves from egg to larva to pupa, and finally to adult. Each stage has a different structure, a different purpose, and even a different way of living in the world. The caterpillar eats leaves. The butterfly may drink nectar. They do not compete with each other, even though they are the same organism at different moments in time. When the caterpillar forms a chrysalis, many of its larval tissues begin to break down. Some muscles that once powered the slow, wave-like motion of crawling are dismantled. Parts of the digestive system, designed for chewing plant matter, are reorganized. Cells undergo a process known as programmed cell death. This is not chaos. It is controlled and guided. The materials released from these tissues are reused, becoming building blocks for the new body that is forming. It also helps to clear up one common confusion. A chrysalis and a cocoon are not the same thing. A chrysalis is the actual pupal body of a butterfly. It is the hardened outer layer of the transforming insect itself. A cocoon, on the other hand, is a silk covering spun by many moth caterpillars around the pupa. Inside a cocoon, there is still a pupa, but the silk layer is an added protective case. Butterflies form chrysalises. Many moths spin cocoons. The transformation inside follows the same basic plan, but the outer wrapping can look very different. If someone were to look inside at just the right moment, the interior might appear soft and partially fluid. This is where the idea of liquefaction began. But the nervous system remains. The breathing tubes, called tracheae, remain largely intact. The imaginal discs remain organized. It is less like melting and more like careful remodeling. Some rooms are taken apart. Others stay standing. New walls are built from familiar material. The transformation does not begin randomly. It is triggered by hormones. As long as a chemical signal called juvenile hormone stays high, the caterpillar continues in its larval state. It eats, it grows, it molts. But when the level of that hormone drops and another hormone called ecdysone rises, the signal changes. Growth as a caterpillar comes to an end. A different program begins. The shift is quiet and chemical. No sound marks it. no light flashes. The change happens within. This strategy, known as holometabolous development, is not rare. In fact, around 80 to 90% of all insect species on Earth undergo complete metamorphosis. Beetles, flies, bees, wasps, moths and butterflies all follow this pattern. It has become one of the most successful biological strategies in evolutionary history. By separating life into distinct stages, insects reduce competition between their younger and older forms. One stage gathers energy, another disperses it. So when we picture a caterpillar resting inside its chrysalis, we are not looking at something that is disappearing. We are looking at something reorganizing. The small body that once clung to the edge of a leaf carries within it a second design. Not visible from the outside, not hurried, simply waiting for the right chemical moment to unfold. This first form was never meant to last, and that is part of its quiet precision. That first form was never meant to last. It was built for one purpose and one purpose only: to gather, to accumulate, to prepare. A caterpillar's life is often described as simple, but it is intensely focused. From the moment it hatches from its egg, its primary task is eating. Tiny mandibles begin cutting into the leaf beneath it. The edge of the leaf slowly changes shape, small crescent bites appearing one after another. What looks like quiet grazing is, in fact, rapid growth in motion. During the larval stage, a caterpillar can increase its body mass thousands of times over. Some species grow more than 1,000-fold between hatching and pupation. This expansion does not happen smoothly in one continuous stretch. It happens in steps, in stages called instars. Between each stage, the caterpillar molts, shedding its outer skin because the exoskeleton cannot stretch indefinitely. Beneath that soft outer layer is a body designed for efficiency. Much of the internal space is occupied by the digestive system. The gut of a caterpillar is proportionally large, running almost the length of its body. Leaves enter. Enzymes break down plant tissue. Nutrients are absorbed and moved into storage. The process is steady and repetitive, like waves passing through a shoreline. Energy gathered from leaves is stored in structures known as fat bodies. These are not like the fat deposits we might picture in mammals. They are specialized tissues that store nutrients, proteins, and lipids. They act as a reserve, a quiet savings account, for a future stage that cannot eat. When the caterpillar later seals itself inside a chrysalis, it will rely entirely on what has been stored during this period of feeding. The caterpillar does not yet know that wings are coming, but its biology is already preparing for them. The imaginal discs mentioned earlier remain small and inactive during much of the larval stage. They do not interfere with feeding or movement. They simply exist within the body, held in place among muscles and digestive tissues, waiting for a different hormonal environment. Growth during this stage is guided by chemical signals. After a period of feeding, levels of ecdysone rise, triggering a molt. The old exoskeleton splits. The caterpillar wriggles free. Its new outer layer is initially soft and expandable. It takes in air, expanding slightly before the new cuticle hardens. Each molt allows another leap in size. Each instar represents a new version of the same temporary design. Even the movement of a caterpillar reflects this stage of accumulation. Without bones, it relies on a hydrostatic skeleton. Fluid within its body provides resistance, allowing muscles to contract in waves from back to front. The body grips the leaf with prolegs equipped with tiny hook-like structures called crochets. The motion is steady, deliberate. It moves only as much as necessary to reach more food. There is a quiet logic to this phase. The larva does not compete with its future adult self. It occupies a different niche. While the caterpillar remains close to leaves, often hidden among green surfaces, the adult butterfly will later rise into open air, visiting flowers and carrying pollen. By separating feeding and reproduction into different stages, the species avoids internal competition. One form gathers energy, the other distributes genes. From the outside, the caterpillar appears unchanged from day to day. It crawls, it eats, it rests. Yet inside, mass is increasing. Nutrients are being stored. Imaginal discs remain in place, silent but intact. Hormonal levels fluctuate in measured cycles. The body that seems so simple is performing one of the most focused acts in the natural world. Preparation without awareness. Soon, feeding will slow. The rhythm of biting and chewing will begin to fade. But for now, the leaf continues to lose its edges, and the larval body continues to expand, gathering everything it will need for a transformation that has not yet begun. The body that gathers so steadily is not meant to be permanent. It is a structure built for a phase, shaped by a single task, and held together just long enough to complete it. If you look closely at a caterpillar, its form appears simple, almost uniform, a long cylinder divided into soft rings. Most caterpillars have 13 distinct body segments. The first 3 segments, just behind the head, each carry a pair of true legs. These are jointed and more rigid, similar in basic design to the legs the adult insect will later use. Behind them are several pairs of fleshy extensions called prolegs. These are not true legs. They are temporary supports, designed only for the larval stage. Along the underside of those prolegs are tiny, hook-like structures known as crochets. Under magnification, they look like curved claws arranged in small circles. These hooks allow the caterpillar to grip the surface of a leaf securely. When it moves, it releases and reattaches these grips in sequence, creating the familiar wave-like motion. It is a slow rhythm, the body arching and extending, inch by inch across the green surface. At the front of the body is a hardened head capsule. This part feels firmer because it is reinforced with thicker cuticle. On that head are strong mandibles, designed for cutting plant tissue. The mouthparts are not built for sipping nectar or tasting flowers. They are blunt, efficient tools for chewing. When the mandibles close against the edge of a leaf, they slice through plant cells and fibers with steady pressure. On either side of the head are small, simple eyes called ocelli. These are not capable of forming detailed images. They detect light and dark, helping the caterpillar orient itself towards shade or shelter. The world, as experienced through these eyes, is likely a blend of brightness and shadow rather than shape. The caterpillar does not see the leaf as we do. It senses its presence through contact, through chemical signals, and through light intensity. Running along the sides of the body are tiny openings called spiracles. These are entry points to the tracheal system, a network of air-filled tubes that deliver oxygen directly to tissues. There are no lungs. Air moves through branching tubes, reaching muscles and organs without the need for a circulatory exchange like in mammals. Even in this temporary stage, the respiratory system is precise and efficient. Beneath the outer skin, muscles are arranged in bands that contract in coordinated waves. There are no bones, but the body maintains shape through internal fluid pressure. This is known as a hydrostatic skeleton. When muscles on one side contract, the internal fluid resists compression, allowing the body to bend and extend. The caterpillar's slow crawl is powered by this internal balance of pressure and contraction. The outer covering, called the cuticle, is flexible but limited. It protects the body and prevents excessive water loss, but it cannot stretch indefinitely. This limitation is what makes molting necessary. Each time the body grows too large for its cuticle, the skin must be shed and replaced. The very architecture of the caterpillar ensures that change will come. It is built into the design. Some species carry small hairs or spines along their bodies. These may serve as deterrents to predators or as sensory structures that detect touch and vibration. In certain species, these hairs can even deliver mild toxins. But even these defenses are part of a temporary plan. The adult butterfly or moth will not retain this same protective strategy. When you picture the caterpillar moving across a leaf, you are seeing a body optimized for gathering and holding energy. Every segment, every hook, every chewing motion serves that aim. It is not designed for flight. It is not designed for reproduction. It is not even designed to last. Inside this soft, segmented architecture, the imaginal discs remain tucked among muscles and digestive tissues. They are quiet passengers within a body that is still focused on eating. The tracheal tubes continue to deliver oxygen. The spiracles open and close gently. The mandibles continue their steady work. From the outside, nothing suggests that this structure will one day dissolve and reorganize. The prolegs cling firmly to the leaf. The head lowers to take another bite. The body arches and extends. Yet every part of this design carries the understanding that it is a phase. The architecture is real. The growth is real. The chewing, the gripping, the breathing, all of it is functional and complete. And still, it is provisional. Soon, the signals within will shift again. The body that now seems stable will reveal itself to be a draft, not a final copy. The body that seems so steady on the surface is already familiar with change. It has been practicing it quietly from the beginning. Because the caterpillar's outer covering cannot stretch forever, growth happens in steps. The cuticle, that soft but protective outer layer, is flexible only to a point. As the body beneath it expands with stored nutrients and growing tissues, pressure builds. The outer layer grows tight. Movement feels slightly constrained. Feeding may slow for a short time, and then, guided by internal signals, the process of molting begins. This process is called ecdysis. It is not sudden or chaotic. It is coordinated and chemical. Levels of a hormone called ecdysone rise in the caterpillar's hemolymph, the fluid that circulates within its body. That hormonal signal instructs the epidermal cells beneath the cuticle to begin separating from the old outer layer. A new cuticle forms underneath, soft and folded, waiting for its moment. If you were watching closely, you might see the caterpillar become still. The body tightens slightly. A faint line appears near the head capsule. Then the old cuticle splits. The caterpillar wriggles forward, slowly pulling itself free from the outer skin that no longer fits. The process can take minutes or hours, depending on the species. What remains behind is a thin, translucent casing, the shape of the former body, empty now. Beneath it, the new cuticle is pale and soft. In this brief window, the caterpillar takes in air, expanding slightly. The body grows into the extra space provided by the new outer layer. Gradually, the cuticle hardens. The segments regain their firmness. The prolegs regain their grip. Feeding resumes. Each of these stages between molts is called an instar. Most caterpillars pass through 4 or 5 instars before reaching full larval size. With each instar, the body becomes larger, thicker, and heavier. The head capsule grows proportionally. The mandibles strengthen. The digestive system continues its work with increasing efficiency. Molting is not only about size, it is also rehearsal. Each shedding of the skin is a controlled transformation. The old exterior is left behind. A new one takes its place. The body proves that it can dismantle and rebuild its outermost layer while preserving internal order. This repeated pattern lays the groundwork for the final and more dramatic change that will come later. The hormonal control behind molting is precise. As long as juvenile hormone remains present in sufficient amounts, the caterpillar molts into a larger larval form. The body retains its caterpillar identity. The imaginal discs remain small and inactive. The digestive tract continues to dominate the internal space. Each molt in this stage leads to another caterpillar, only larger. But the chemistry carries a clock within it. Over time, as the caterpillar approaches its maximum larval size, the balance begins to shift. Juvenile hormone levels gradually decrease. The body becomes more sensitive to ecdysone. The next molt will not simply produce a larger version of the same design. It will respond differently. From the outside, these earlier molts may look like small adjustments. A caterpillar that was once barely visible on the leaf becomes thicker, its segments more pronounced. The leaf beneath it shows larger missing sections where bites have been taken. The motion across the surface remains steady, but the weight pressing into the leaf has increased. Even during these repeated molts, the imaginal discs remain present. They are not yet expanding into wings or antennae, but they grow slowly in proportion with the body. They are carried through each instar, protected during each shedding. The blueprint survives every discarded skin. Molting also refreshes the cuticle's protective functions. The new outer layer reduces water loss and restores flexibility. It allows the spiracles along the sides of the body to continue opening and closing efficiently. The hydrostatic skeleton regains full responsiveness. Muscles once constrained by a tight outer layer now move freely again. When you picture the caterpillar lifting the front of its body and inching forward, gripping and releasing with careful rhythm, you are seeing a body that has already changed several times. It is not the same body that first emerged from the egg. It has shed its outer self again and again, each time revealing a slightly different version. These repeated cycles are gentle rehearsals for a larger transition. The act of leaving one form behind has already become familiar. The caterpillar does not know that its final molt will reveal something entirely new, but its biology has been preparing for that possibility from the very beginning. For now, another instar begins. The leaf continues to thin at its edges. The body continues to expand, and the quiet pattern of shedding and renewal moves forward, one layer at a time. Each of those molts followed a familiar pattern. The outer layer split. A larger caterpillar emerged. The rhythm repeated itself with calm predictability. But eventually, the pattern changes. The same hormones are present, yet their balance shifts. And that shift alters the outcome. Inside the caterpillar's body, transformation is guided by chemistry. Two hormones play central roles. One is called juvenile hormone. As long as it remains at a certain level, the caterpillar continues being a caterpillar. It feeds, it grows, it molts into a slightly larger version of itself. The presence of juvenile hormone acts like a signal that says, continue this phase. The second hormone is ecdysone. It rises before each molt, triggering the shedding of the cuticle. During the early instars, when juvenile hormone is still abundant, the surge of ecdysone produces another larval stage. The outer skin splits, and a larger caterpillar steps forward. But over time, the concentration of juvenile hormone declines. This decrease does not happen all at once. It gradually lowers as the caterpillar approaches its maximum size. When ecdysone rises again in an environment where juvenile hormone is no longer dominant, the result is different. The same molting signal now produces a new outcome. Instead of forming another caterpillar, the body begins the process of pupation. It is not a dramatic signal. There is no sudden change in color or shape at first. The caterpillar may still be resting on a leaf. It may still be gripping the surface with its prolegs. Yet internally, gene expression patterns begin to shift. Cells that were once dedicated entirely to growth and digestion start responding to a different set of instructions. Hormones travel through the hemolymph, reaching tissues throughout the body. In response, certain genes are activated while others are silenced. The imaginal discs, which have remained quiet passengers during the feeding phase, begin preparing for expansion. They respond to the new hormonal environment by increasing cell division. The blueprint that was held in reserve becomes active. Meanwhile, the digestive tract, once the dominant internal structure, prepares to be remodeled. Muscles that powered crawling begin to change their protein composition. Even the metabolism adjusts. The body transitions from rapid intake and growth to conservation and restructuring. From the outside, this hormonal shift may appear subtle. The caterpillar's appetite often begins to decrease. The steady biting along the leaf's edge slows. Instead of feeding continuously, it may pause for longer intervals. There is a slight restlessness. It may move away from its usual feeding site. These behavioral changes are outward reflections of the internal chemical transition. The hormonal switch is precise. If juvenile hormone levels remain artificially high in experiments, the caterpillar can be forced to molt into oversized larval forms rather than pupate. If juvenile hormone drops too soon, development can be disrupted. The timing must be exact. Evolution has refined this balance over millions of years, tuning the chemical signals to environmental cues such as temperature and day length. In this way, metamorphosis is not an accident. It is a regulated sequence. Hormones function as messengers, carrying instructions through fluid pathways. Cells listen. Tissues respond. The body changes course not through awareness, but through molecular conversation. You can imagine the caterpillar resting on the underside of a leaf, the green surface filtering soft light from above. Its spiracles open and close gently along its sides. Inside, hormone levels shift like tides. Signals rise and fall. The same chemical that once triggered simple growth now prepares the body for something entirely different. The caterpillar does not decide to transform. It responds. The drop in juvenile hormone removes the instruction to remain a larva. The next pulse of ecdysone no longer means grow larger. It means become something else. The pattern that once produced repetition now produces divergence. And with that quiet chemical change, the path toward the chrysalis begins. The steady rhythm of biting and chewing does not continue forever. At a certain point, the leaf beneath the caterpillar no longer loses its edge in the same patient way. The mandibles pause more often. The body remains still for longer stretches. Appetite, once constant, begins to fade. This slowing is one of the earliest outward signs that metamorphosis is near. Internally, hormone levels have already shifted. Juvenile hormone has dropped to a level that no longer supports continued larval growth. Ecdysone has begun preparing the tissues for a different outcome. But from the outside, what we first notice is behavioral. A caterpillar that once spent most of its waking time feeding begins to wander. Instead of staying on the host plant where it hatched and grew, it moves away from familiar leaves. The hydrostatic waves that once carried it from one feeding site to another now carry it across stems, along branches, sometimes down toward the ground or up toward a sheltered underside. This wandering phase is not random. It is purposeful, though guided entirely by internal programming. The caterpillar is searching for a suitable location to pupate. The site must offer protection from predators, stability for attachment, and the right environmental conditions. Too exposed, and it may be seen by birds. Too damp, and fungal growth could threaten development. Too unstable, and the structure it forms may fall before transformation is complete. During this time, feeding decreases dramatically. The digestive tract, once dominant, begins preparing for dismantling. There is little benefit in consuming more leaves now. The energy required for transformation has already been stored in fat bodies. Nutrients gathered over weeks of constant feeding will now sustain the organism through a stage in which it cannot eat at all. The body itself begins to change in subtle ways. The caterpillar may empty its gut, clearing waste material. This reduces internal mass and makes the body more compact. The color may shift slightly in some species, signaling that pupation is imminent. Muscles that once powered crawling begin responding differently to hormonal signals. Movement becomes more deliberate, less exploratory. If you picture the caterpillar now, you might see it moving slowly along a branch rather than the broad surface of a leaf. The prolegs grip the bark or stem. The head lifts and lowers as if testing the surface. It may circle an area several times before settling. This restlessness reflects internal change. The hormonal cascade that will soon trigger the final molt is already underway. Selection of a pupation site varies by species. Some butterflies attach themselves to the underside of leaves. Others climb to fences, walls, or tree trunks. Moth caterpillars may burrow into soil or spin silk among leaf litter. Despite these differences, the guiding principle is the same. Find a place where stillness can be maintained. The decrease in appetite and the onset of wandering represent a transition from accumulation to preparation. For weeks, the caterpillar's biology was oriented toward growth. Now it shifts toward conservation and transformation. The body that once expanded rapidly begins to contract slightly. The outer segments draw closer together. The motion across surfaces becomes slower, almost measured. Internally, gene expression patterns continue to shift. Cells receive hormonal instructions that alter their function. Some tissues begin downregulating growth-related pathways. Others begin preparing for structural reorganization. The imaginal discs, still small but present, are poised to respond fully once the final molt occurs. From the outside, the caterpillar's stillness can look like fatigue. But it is not weakness. It is a recalibration. The body is preparing to enter a stage where movement will cease entirely. The wandering ends not in collapse, but in anchoring. Soon, the caterpillar will stop moving altogether. It will attach itself securely to a chosen surface. The body will suspend between past and future forms. An external stillness will begin to reflect the deeper internal transformation already in motion. The wandering slows, and then it stops. The search has ended. The caterpillar has chosen its place. What comes next is not dramatic. There is no sudden transformation yet. There is only preparation for stillness. The body, which has moved steadily across leaves and branches for weeks, begins producing silk from specialized glands near the mouth. This silk is a protein-based fiber, liquid inside the body and solidifying when exposed to air. It emerges as a fine strand, almost invisible unless light catches it at the right angle. The caterpillar uses this silk first as an anchor. It may attach itself to the underside of a leaf, to a twig, or to the rough surface of bark. In butterflies, a small silk pad is laid down, and the rear end of the body grips this pad securely, using tiny hook-like structures. Some species also spin a thin silk girdle around the midsection, looping it over the body like a support strap. The silk is strong relative to its thickness, capable of holding the entire weight of the larval body without strain. Once attached, the caterpillar's posture changes. In many butterfly species, the body curls into a distinctive J shape. The head points downward. The segments compress slightly. The prolegs that once gripped leaves now relax, leaving the body suspended by the rear attachment. Movement becomes minimal. The slow waves of crawling cease. This suspension marks a threshold. Externally, the caterpillar appears quiet, perhaps even vulnerable. Internally, hormone levels are rising again. Ecdysone circulates through the hemolymph, reaching cells that are already primed for change. The drop in juvenile hormone has removed the instruction to remain a larva. The next molt will not produce another caterpillar. The body shortens slightly during this phase. muscles contract in subtle adjustments. The cuticle tightens around the form. If you look closely, the outer skin may appear duller, less flexible. The digestive system, already mostly emptied, no longer dominates the internal space. The stored nutrients within the fat bodies are now reserved for reconstruction. Stillness, at this stage, is active. Though the caterpillar is no longer feeding or wandering, cellular communication continues throughout the body. Signals pass from endocrine tissues to target cells. Genes associated with larval growth are being downregulated. Genes required for pupal development are being activated. The imaginal discs begin responding more fully to the hormonal environment, preparing to expand once the final molt occurs. The silk attachment is not merely mechanical support. It ensures stability during a vulnerable moment. When the old cuticle splits, the body must remain steady. Any fall during this transition could be fatal. Evolution has refined this anchoring behavior so that the transformation can proceed without interruption. If you picture the suspended caterpillar now, hanging in its J shape, you see a body that seems paused. The spiracles along its sides still open and close gently, allowing air to move through the tracheal system. The heart-like dorsal vessel continues pumping hemolymph. Oxygen flows. Nutrients circulate. Though movement has ceased, life remains fully active within. In some moth species, the process differs slightly. Rather than hanging openly, they spin a more enclosed silk structure, forming a cocoon. This cocoon may incorporate bits of leaf, soil, or bark, creating camouflage. But even in these cases, the principle is similar. Secure the body, reduce exposure, prepare for immobility. The J-shaped posture in butterflies is a visible signal that the final larval stage has ended. It is a bridge between forms. The prolegs that once gripped surfaces so reliably now release their hold. The body's balance shifts entirely to the silk anchor. Gravity pulls gently downward, maintaining tension in the attachment. Inside, the cuticle begins to separate once more from the epidermal layer beneath. A new form is already developing underneath the old skin. Unlike previous molts, this one will not reveal a larger caterpillar. It will reveal a chrysalis. For a brief time, the caterpillar remains suspended, still and quiet. From the outside, it looks almost unchanged. Yet beneath the familiar segmented exterior, the next structure is ready. The attachment is secure. The posture is set. The chemical signals are aligned. External stillness now reflects internal readiness. Soon, the outer skin will split one final time, and the temporary architecture of the caterpillar will give way to something entirely new. The body hangs in its J shape, motionless except for the faint movement of air through spiracles. From the outside, it still looks like a caterpillar. The segments are familiar. The texture of the cuticle is unchanged. But beneath that outer layer, a different structure has already formed. The final molt begins quietly. As before, ecdysone levels rise, signaling that it is time for the cuticle to separate from the epidermal cells beneath it. The old larval skin loosens. A faint line appears near the head capsule. The body contracts in subtle pulses, and then the cuticle splits. What emerges this time is not another caterpillar. The outer larval skin peels back, inch by inch, as the organism wriggles within its silk anchor. The process is deliberate. The old head capsule slides off. The familiar segments of the caterpillar compress and shift. Beneath the shedding skin, a hardened, smooth surface is revealed. The shape is no longer segmented in the same way. The prolegs disappear from view. The overall form becomes more compact, more enclosed. This newly exposed structure is called a chrysalis in butterflies. In moths, the organism may be enclosed in a cocoon made of silk, but the underlying pupal form is similar. The chrysalis is not an additional layer added on top of the caterpillar. It is the pupa itself, the next stage in development. As the last of the larval skin slides free, it often falls away or remains briefly attached before dropping. What hangs in its place is still. The surface of the chrysalis may be smooth, sometimes angular, sometimes adorned with small ridges or subtle metallic flecks, depending on the species. The color may range from green to brown, often matching the surrounding environment for camouflage. Immediately after the larval skin is shed, the chrysalis is soft. The outer covering, known as the pupal cuticle, must harden. Over the next several hours, proteins within the cuticle undergo chemical changes that increase rigidity. This process, called sclerotization, strengthens the outer casing. The once flexible larval body is now encased in a protective shell. The shift in external form is immediate and striking. Where there were once visible legs and mandibles, there is now a sealed structure. The segmentation of the caterpillar is no longer apparent in the same way. The organism has entered a stage designed for immobility. It is important to distinguish between a chrysalis and a cocoon. A chrysalis is the pupal stage of butterflies, and it is the hardened outer layer of the pupa itself. A cocoon, by contrast, is a silk structure spun by many moth caterpillars around the pupa. Inside the cocoon, there is still a pupa, but the cocoon is an additional protective casing made of silk. In butterflies, the pupa is exposed, attached only by silk at the rear, or supported by a silk girdle. If you picture the chrysalis now, hanging quietly beneath a leaf or against a branch, it appears calm and self-contained. The spiracles remain present along its surface, allowing gas exchange to continue. Air still flows through the tracheal system. The dorsal vessel continues to circulate hemolymph. Though the exterior is rigid, internal life has not paused. The transformation from caterpillar to pupa marks the final disappearance of the larval architecture. The chewing mandibles are gone. The prolegs are no longer visible. The body has sealed itself into a shape that hints at what will come, but does not yet reveal it fully. Within the hardened casing, tissues that once powered crawling are preparing for dismantling. Imaginal discs are positioned to expand. Hormonal signals continue their work, guiding gene expression in precise patterns. The environment outside may remain unchanged. The same branch, the same filtered light through leaves. But inside, the biological program has shifted entirely. From this moment forward, the organism will not move across surfaces. It will not eat. It will not wander. The chrysalis represents commitment to transformation. The outer skin that once defined the caterpillar has been left behind. In its place hangs a structure built for enclosure and change. What appears still from the outside now contains the beginning of internal dismantling. And within this sealed form, the next phase begins to unfold. The chrysalis hangs quietly, its surface firm and unmoving. From the outside, it appears sealed and complete. But inside, the most delicate phase of transformation has just begun. The larval body that once filled the interior space does not remain intact. Soon after the final molt, many of the caterpillar's tissues begin to break down. Muscles that once powered the slow arching crawl across leaves are dismantled. The long digestive tract, built for constant chewing and processing of plant material, begins to shrink and reorganize. This is not sudden collapse. It is guided and precise. Cells follow instructions encoded within their genes. Some undergo apoptosis, a process often described as programmed cell death. Unlike injury or decay, apoptosis is orderly. Cells dismantle themselves in controlled steps. Their contents are broken down into smaller components. Proteins and nutrients are released into the internal environment, where they can be reused. Nothing is wasted. As these larval tissues dissolve, the interior of the chrysalis can appear soft and partially fluid. This is where the myth of total liquefaction began. At certain moments during development, the internal environment does become semi-fluid in consistency. Yet even here, structure remains. The nervous system largely persists. The tracheal tubes that deliver oxygen continue functioning. The imaginal discs remain intact and organized. It is less like melting and more like selective remodeling. Some structures are carefully taken apart. Others are preserved as scaffolding. The hemolymph continues to circulate through the body, carrying nutrients from dismantled tissues to growing ones. Oxygen flows through the spiracles along the outer casing. The organism is not dissolving into chaos. It is reallocating resources. Within this sealed chamber, the balance between breakdown and rebuilding is tightly regulated. Hormones continue to influence gene expression. Cells respond differently depending on their location and developmental role. Larval muscle fibers fragment and release their stored proteins. Meanwhile, precursor cells destined to form adult flight muscles begin increasing in number. The digestive tract offers a clear example of this transformation. In the caterpillar, the gut is long and specialized for processing leaves. In the adult butterfly, the digestive system is redesigned for liquid intake. The larval gut must be reshaped into a narrower, more delicate structure suited for nectar. This reorganization requires dismantling of old tissues and growth of new ones. Throughout this process, the imaginal discs begin expanding more actively. These clusters of cells, which have remained dormant during the larval stage, now receive strong hormonal signals to proliferate. They fold and differentiate, forming the outlines of wings, legs, antennae, and compound eyes. As larval tissues break down around them, the imaginal discs grow into the spaces created. The internal environment may briefly appear disordered if viewed at the wrong moment. But beneath that softness is coordination. Molecular signals ensure that adult structures form in precise positions. Cells communicate through chemical gradients. Proteins are synthesized according to newly activated genetic programs. The blueprint that was carried silently within the caterpillar now unfolds in 3 dimensions. If you imagine the chrysalis suspended beneath a leaf, its surface still and unchanging, you might picture the interior as dark and silent. Yet inside, microscopic construction is underway. Old fibers are being disassembled. New tissues are weaving themselves into place. The dorsal vessel continues to pulse gently. Spiracles open and close, allowing air to circulate. The breakdown of larval tissues provides raw materials for rebuilding. Amino acids, lipids, and sugars released during apoptosis are transported through the hemolymph to areas of active growth. In this way, the energy gathered during weeks of feeding is redirected. The leaves consumed earlier now fuel the formation of wings. The semi-fluid phase does not last indefinitely. As reconstruction advances, the interior becomes more structured again. Adult muscles attach to developing exoskeletal elements. Organs settle into defined positions. What began as dismantling gradually shifts toward organization. From the outside, nothing announces this internal shift. The chrysalis remains attached, quiet against the branch. Yet within its protective casing, the larval body is being carefully replaced. Not erased, but transformed through controlled dismantling. The myth says the caterpillar turns into liquid. The reality is more intricate. It is a period of selective dissolution, guided by precise biological signals, preparing space for something new to take shape. And at the center of that emerging design are the imaginal discs, ready to build. Within the soft interior of the chrysalis, as larval tissues are carefully dismantled, certain structures remain untouched by that wave of dissolution. They are small, organized clusters of cells, positioned in specific places along the body. They have been present since the earliest stages of development, and now, at last, they begin to unfold. These clusters are called imaginal discs. Long before the caterpillar spun silk or took its first bite of leaf, these discs were already there. They formed during embryonic development, when the organism was still inside its egg. At that stage, the genetic blueprint for both larval and adult structures was established. The imaginal discs were set aside early, remaining dormant while the larval body carried out its temporary task of growth. During the weeks of feeding and molting, these discs stayed small. They did not interfere with the caterpillar's chewing mandibles or its prolegs. They did not form visible wings. They remained compact, layered sheets of cells, folded within the body like quiet maps waiting to be opened. Now, inside the chrysalis, hormonal signals activate them fully. In response to rising levels of ecdysone and the absence of juvenile hormone, the cells within each imaginal disc begin dividing rapidly. They do not grow randomly. Their expansion follows precise genetic instructions. Each disc corresponds to a particular adult structure. There are discs for the wings, for the legs, for the antennae, for the compound eyes, and for portions of the thorax and abdomen. As the surrounding larval tissues break down and release nutrients, the imaginal discs use those resources to fuel their growth. Proteins and amino acids circulate through the hemolymph and are absorbed by the proliferating cells. The space created by the dismantling of larval muscles and gut tissue becomes a framework in which new structures can expand. If you could see inside at this stage, you would not observe fully formed wings yet. Instead, you would see sheets of cells thickening and folding. The wing discs flatten and elongate, establishing the future pattern of veins and scales. Leg discs extend outward, differentiating into segments that will later articulate as joints. Eye discs begin shaping the compound structure composed of hundreds or thousands of individual lenses. Each cell within these discs carries the same genetic material. What determines its fate is position and chemical signaling. Cells at one edge of a disc may receive a slightly different concentration of signaling molecules than cells at another edge. These gradients guide specialization. Some cells become part of a wing membrane. Others become veins that provide support and carry hemolymph. Others develop into sensory structures. The organization is layered and deliberate. It is not a formless mass solidifying into shape. It is patterned growth, unfolding from a pre-existing template. The imaginal discs were never passive passengers. They were placeholders, holding spatial information that would only become visible at this stage. Meanwhile, the nervous system adjusts to accommodate these new structures. Nerve connections extend toward developing appendages. Muscles forming from precursor cells align with attachment points on the emerging exoskeleton. The adult form is not simply being assembled piece by piece. It is being integrated. Throughout this expansion, the chrysalis remains outwardly still. The hardened cuticle shields the interior from mechanical disturbance. Oxygen continues to diffuse through spiracles into the tracheal network. The dorsal vessel pulses, distributing nutrients and removing waste. The entire transformation unfolds in a sealed environment where internal communication replaces movement. It is striking to consider that the blueprint for wings existed within the caterpillar all along. While it crawled across leaves, gripping with prolegs and chewing plant fibers, the potential for flight was already encoded within these discs. The larval body carried within it a second design, waiting for the correct hormonal signal to activate it. As the imaginal discs expand and differentiate, the semi-fluid quality of the interior gradually gives way to structure again. New exoskeletal elements begin forming. Tissues attach in coordinated arrangements. What was once a breakdown phase transitions into active construction. Inside the chrysalis, the quiet builders have begun their work in earnest. The larval architecture recedes. The adult plan advances. And the hidden blueprint, present since the beginning, unfolds into visible form. As the imaginal discs expand and begin shaping wings, legs, and antennae, the interior of the chrysalis becomes less fluid and more defined. What was once a period of selective dismantling now shifts steadily toward reconstruction. The process is not hurried. It unfolds in layered stages, guided by cellular signals that move quietly from one tissue to the next. The adult body does not simply appear fully formed. It is assembled piece by piece within the protective casing. The digestive system, once long and built for chewing leaves, continues its transformation. In the caterpillar, the gut was wide and specialized for breaking down tough plant fibers. In the adult butterfly, the digestive tract becomes narrower and more suited to liquid intake. The muscular structure changes. The lining adapts. Enzymes that were once optimized for leaf digestion give way to systems prepared for nectar. At the same time, new muscle groups begin forming. The most significant of these are the flight muscles located in the thorax. During the larval stage, The caterpillar's muscles were arranged for crawling, contracting in waves along the segmented body. Now, precursor cells proliferate and differentiate into strong bundles designed for rapid wing movement. These muscles must anchor securely to the developing exoskeleton. Their attachment points are carefully aligned as the thoracic structure takes shape. The exoskeleton itself reorganizes. In the caterpillar, the outer cuticle was soft and segmented, allowing flexibility. In the adult form, the exoskeleton must support flight and protect delicate organs. Plates harden and join in new configurations. The segmentation remains, but its function shifts. Instead of aiding crawling, it supports articulation of wings and legs. Throughout this process, the nervous system remains continuous. It does not dissolve completely during the earlier dismantling phase. Instead, it adapts. Nerve pathways extend into the developing appendages. Signals that once coordinated crawling will soon coordinate wing beats. Sensory systems also mature. Compound eyes, formed from imaginal discs, differentiate into hundreds of small lenses called ommatidia. These lenses will allow the adult to detect movement and light patterns far more complex than the simple light and dark perception of the larval ocelli. The tracheal system, responsible for delivering oxygen directly to tissues, undergoes modification as well. The branching air tubes that once served a crawling body adjust to meet the metabolic demands of flight. Oxygen consumption will increase dramatically once the adult begins flying. The tracheae expand and reorganize to supply developing flight muscles efficiently. Hemolymph continues circulating through the body, carrying nutrients released during earlier tissue breakdown. As new structures solidify, the flow patterns adjust. The internal environment becomes more structured, less like a construction site and more like a finished interior slowly revealing itself. If you imagine the chrysalis now, its surface still and sealed. The internal space has changed significantly since the first days of pupation. Where there was once softness and partial dissolution, there are now defined outlines of wings folded tightly against the body. The antennae are present, curled delicately. The legs are compacted but fully shaped. The adult form exists in miniature, waiting for the final stages of strengthening. Gene expression continues to guide this reconstruction. Specific genes regulate pigment production in wing scales. Others control the arrangement of veins within the wings, ensuring structural support and proper flow of hemolymph once expansion occurs. The pattern of coloration that will one day be visible is already being determined at the cellular level. Temperature and environmental conditions can influence the rate of these processes. Warmer temperatures often accelerate development by increasing metabolic activity. Cooler conditions slow cellular division and protein synthesis. Yet the sequence remains ordered. The timing may stretch or compress slightly, but the steps occur in the same progression. From the outside, days may pass without visible change. The chrysalis may darken slightly as pigments develop beneath its surface. In some species, the outline of wings becomes faintly visible through the casing shortly before emergence. But until that moment, the transformation remains concealed. Inside, muscles anchor firmly. Organs settle into their adult positions. The digestive tract completes its reshaping. The nervous system integrates new sensory inputs. The adult insect is nearly complete. What began as a body devoted entirely to feeding has been reorganized into one prepared for flight and reproduction. The reconstruction is nearly finished. Only time and final strengthening remain before the seal casing yields to movement once again. Inside the hardened casing, reconstruction continues in silence. The outline of wings is present. The muscles have formed. The organs have reorganized. And yet, the chrysalis remains sealed, held in place by silk against branch or leaf. What determines how long it remains this way is not only the internal program, but the world outside. Time inside the chrysalis is shaped by temperature. Developmental duration varies widely by species. Some butterflies complete pupation in as little as 1 to 2 weeks. Others require several months. The internal sequence of breakdown and rebuilding follows the same order, but the pace can stretch or compress depending on environmental conditions. Insects are ectothermic, meaning their body temperature is influenced by the surrounding air. When temperatures are warmer, metabolic reactions occur more quickly. Enzymes involved in cell division and protein synthesis operate at higher rates. Hormonal signals are processed faster. The imaginal discs expand more rapidly. Muscles differentiate sooner. The entire transformation proceeds at an accelerated pace. When temperatures drop, everything slows. Inside a chrysalis exposed to cooler air, cellular activity becomes more measured. Chemical reactions that drive tissue formation proceed at a lower rate. Protein assembly takes longer. The dorsal vessel continues to pulse, but circulation may be slower. Oxygen still moves through the tracheal tubes, yet the demand for it is reduced because metabolic activity has decreased. In some climates, development pauses almost entirely. This state is called diapause. Diapause is not simply a response to cold. It is a hormonally regulated suspension of development that allows the organism to survive unfavorable conditions, often winter. During diapause, metabolism slows dramatically. Cell division halts. Growth pauses at a defined stage. The chrysalis may remain intact for months, waiting for environmental cues such as rising temperatures or longer daylight to resume development. This seasonal synchronization ensures that emergence occurs at the right time of year. A butterfly that emerges too early in cold conditions may find no flowers for nectar. One that emerges too late may miss optimal mating opportunities. Through generations of refinement, species have aligned their internal timing with seasonal patterns in their habitat. If you imagine the chrysalis hanging quietly through shifting weather, you might picture warm days followed by cool nights. Inside, development adjusts subtly to each fluctuation. A rise in temperature may slightly increase metabolic rate. A drop may slow it again. Yet the internal program remains intact, advancing only when conditions permit. The metabolic slowdown during pupation serves another purpose. Energy must be conserved. The organism cannot feed during this stage. All the nutrients required for reconstruction were stored during the larval period. By lowering metabolic demands, especially in cooler environments, the chrysalis ensures that energy reserves are not depleted before development is complete. The duration of this stage can also vary within a species. Individuals in warmer regions may complete metamorphosis more quickly than those in cooler latitudes. Some species produce multiple generations per year in warm climates, with shorter pupal durations. In colder regions, there may be only one generation annually, with pupae entering diapause to bridge the winter months. From the outside, days may pass with no visible change. The surface of the chrysalis may gradually darken as pigments develop in the forming wings beneath. In some species, the outline of the wings becomes faintly visible through the casing shortly before emergence. These subtle shifts mark the nearing completion of internal reconstruction. Inside, muscles strengthen. The exoskeleton finishes hardening in its adult configuration. Wing veins finalize their structure. The nervous system refines its connections to newly formed appendages. The body approaches readiness. The stillness of the chrysalis is not emptiness. It is measured progress shaped by temperature, season, and evolutionary timing. Whether days or months pass, the transformation advances in step with the environment. Soon, when the internal form is fully prepared and the external conditions are favorable, the sealed casing will no longer be needed. The quiet pause of pupation will give way to movement once more. Inside the chrysalis, where stillness has been shaped by temperature and time, the adult form approaches completion. The outline of wings, once no more than folded sheets of imaginal tissue, becomes more defined. Structures that will soon catch air are assembled in quiet precision. The wings begin as thin, layered membranes formed from expanded imaginal discs. At first, they are compact and folded tightly along the body. Within them, a network of veins develops. These veins are not decorative. They provide structural support and will later carry hemolymph through the wing as it expands. The pattern of these veins follows a genetically determined map, guiding strength and flexibility in specific regions. Between the veins, cells differentiate into what will become scales. Butterfly wings are covered in thousands of tiny scales, each overlapping like shingles on a roof. These scales are modified hairs, flattened and specialized. Under magnification, they reveal intricate textures and ridges. During development inside the chrysalis, scale precursor cells multiply and arrange themselves across the wing surface in ordered rows. Pigment formation begins at the cellular level. Some pigments are produced chemically within the cells themselves. Others result from microscopic structures that reflect light in specific ways, creating iridescent blues or shifting colors without relying on traditional pigments. The pattern that will later be visible from a distance is encoded in the spatial arrangement of these scales and pigments. Cells at one edge of the wing receive different signals than those at another edge, leading to bands, spots, or symmetrical designs. Symmetry is critical. Both wings must develop in mirror image. Chemical gradients and gene expression patterns ensure that the left and right sides follow corresponding developmental paths. Even slight variations can alter the visual pattern. Yet through millions of generations, the mechanisms guiding symmetry have become remarkably reliable. As the wings form, muscle attachments strengthen within the thorax. The large flight muscles that were constructed during earlier stages now anchor securely to the internal skeleton. These muscles are arranged in bundles that will contract rapidly once the butterfly begins flying. Unlike the slow, wave-like contractions of the caterpillar's crawling muscles, flight muscles must generate quick, repeated movements. Hemolymph channels within the wings develop in coordination with the veins. Though the wings are not yet expanded, their internal pathways are established. Once the adult emerges, hemolymph will be pumped into these channels to unfurl and stiffen the wings. For now, they remain folded, compact, and waiting. The exoskeleton of the adult continues to harden gradually. Plates that will support wing articulation become more rigid. Joints between segments refine their structure. Antennae, formed from imaginal discs near the head region, elongate and coil slightly, preparing for sensory function. The compound eyes complete their differentiation, with each ommatidium forming its lens and light-sensitive cells. Within the chrysalis, oxygen continues to move through spiracles into the tracheal system. As the adult structures solidify, metabolic demand may increase slightly, reflecting the energy required for final assembly. The dorsal vessel pulses steadily, circulating nutrients to areas of active tissue formation. If you imagine the chrysalis suspended against a branch, its outer surface still firm and opaque, you might sense that something inside has reached a threshold. The internal space that once held semi-fluid larval remnants is now organized. Wings are folded in place along the body. Legs are positioned beneath. The proboscis, a long coiled feeding tube, forms from paired structures that fuse and elongate. In many species, as development nears completion, the chrysalis darkens. The once uniform exterior may become slightly translucent. The colors of the wings, still folded and pressed against the inner surface, begin to show faintly through the casing. This subtle shift is one of the few external signs that emergence is approaching. The transformation from leaf-eating larva to winged adult has nearly concluded. Every vein, scale, muscle attachment, and sensory structure has taken shape within the sealed chamber. The adult insect exists now in miniature, fully formed but compressed. Soon, the folded wings will need space. The hardened casing that protected this reconstruction will no longer be necessary. Internal pressure will build. Muscles will contract in new patterns. The structures that have unfolded invisibly are ready to meet air. The chrysalis has held its shape through days or weeks of quiet transformation. Its surface has darkened slightly. The faint outlines of wings may now be visible beneath the casing. Inside, muscles are formed, organs are aligned, and the folded wings are ready. What remains is release. The moment of emergence begins with pressure from within. The adult butterfly, now fully formed but compressed, begins contracting its thoracic and abdominal muscles. These movements are subtle at first, barely perceptible from the outside. Gradually, they increase internal pressure against the pupal casing. The cuticle that once protected the developing form has completed its purpose. A thin seam along the chrysalis weakens. Then, the casing splits. The rupture usually occurs along the dorsal side, near the head and thorax. The hardened shell opens in a controlled crack, rather than an explosive break. The adult pushes outward, slowly working its way free. The head emerges first, followed by the thorax. The legs extend and grip the surface nearby, often the same branch or leaf to which the chrysalis was attached. The wings at this moment are not yet recognizable as the wide, symmetrical structures associated with butterflies. They are small, crumpled, and folded tightly against the body. The entire insect appears compact and slightly soft. The exoskeleton, though formed, has not fully hardened. Once free from the casing, the butterfly hangs downward. Gravity assists in the next crucial step. Hemolymph begins pumping forcefully through the network of veins inside the wings. The dorsal vessel increases circulation, and muscles in the abdomen contract rhythmically to help drive fluid into the wing channels. As hemolymph fills the veins, the wings begin to expand. The crumpled membranes stretch outward. Veins become more visible as they stiffen with internal pressure. The surface area increases gradually, unfolding from its compressed state. This process can take several minutes to an hour, depending on species and environmental conditions. During this time, the butterfly remains still, allowing fluid to distribute evenly. The expansion is precise. Each vein guides the unfolding pattern. The symmetry established during development ensures that both wings extend in mirror image. The scales that formed inside the chrysalis now lie flat across the surface, revealing color and pattern for the first time. As the wings reach their full size, the hemolymph begins to recede slightly from the outermost regions. The fluid that served to expand the structure now supports initial rigidity, while the cuticle undergoes final hardening. This process, called sclerotization, strengthens the exoskeleton of both the body and wings. Proteins within the cuticle cross-link chemically, increasing durability. During this drying phase, the butterfly remains vulnerable. The wings must dry fully before flight. Any disturbance at this stage can cause deformities. Wind, predators, or physical contact could interfere with proper expansion. For this reason, emergence timing is often aligned with favorable environmental conditions, such as calm weather and moderate temperatures. The empty chrysalis remains attached nearby, its purpose fulfilled. It may hang open and translucent, a light shell beside the newly emerged adult. The transformation from larva to winged insect is now visible in complete form. The antennae extend and adjust, sampling the air. The proboscis, which developed as two separate halves inside the chrysalis, must be joined. The butterfly curls and uncurls it repeatedly, allowing the two halves to fuse into a single functional tube capable of drawing nectar. Gradually, the wings stiffen. Their surface becomes firm enough to resist bending under their own weight. The muscles within the thorax begin small test contractions, flexing the wings gently. Sensory systems, including compound eyes and antennae, are now fully operational. What was once a leaf-bound crawler now hangs poised above open air. The moment of emergence is not rushed. It is deliberate and careful. The shell has given way. The compressed form has expanded. The final strengthening continues quietly. Soon, the wings will lift for the first time. The first flight does not happen right away. After the wings expand and dry, the butterfly stays still for a while. It must learn how to use a body that is completely different from the one it had before. For weeks, the caterpillar moved by crawling. Its body bent in waves. Tiny legs called prolegs gripped leaves and branches. Its world was close to the ground, measured in inches. Now everything has changed. The butterfly lifts its wings slowly. The movement is careful at first. The large muscles inside the chest tighten and release. The wings rise and fall in small test beats. Air moves across the surface of the scales. The body feels lighter than it ever has before. Crawling and flying are not just different actions. They require different coordination. The nervous system, which stayed active during the time inside the chrysalis, must now guide new patterns of movement. Signals that once helped the caterpillar bend its body now help the butterfly balance in the air. The first full wing beats are stronger. The butterfly grips the branch tightly with its thin legs. Then it loosens its hold. For a brief moment, it lifts off. The air feels different from the surface of a leaf. It pushes back against the wings. The butterfly adjusts its angle. If the wind is calm, it may glide a short distance before landing again. The first flights are often short. They are practice. Vision plays a big role in this new way of moving. The caterpillar had simple eyes that could sense light and dark. The butterfly now has compound eyes made of many small lenses. These lenses work together to detect movement and color. The world is brighter and more detailed than before. The antennae also guide the butterfly. They can sense smells carried on the breeze. They help with balance and direction. When the butterfly turns, the antennae shift slightly, gathering information from the air. Feeding changes too. The chewing mouthparts of the caterpillar are gone. In their place is a long tube called a proboscis. It is usually curled up under the head like a spring. When the butterfly finds a flower, it uncurls the proboscis and reaches into the center to drink nectar. Learning to use the proboscis takes practice. The butterfly may land on several flowers before feeding successfully. It tests its footing with its legs. It senses sweetness with receptors on its feet. Then it lowers its head and extends the tube. The relationship to space is completely new. The caterpillar lived on one plant or a small group of plants. The butterfly can travel across fields, gardens, and forests. It can rise above obstacles. It can follow sunlight. With flight comes distance. With distance comes choice. The butterfly is no longer tied to a single leaf. It can search for nectar in many places. It can look for a mate across open land. It can avoid some predators by lifting away into the air. Yet flight also brings new risks. Birds can see a butterfly more easily than they can see a caterpillar hidden among leaves. The bright colors that attract mates may also attract hunters. Survival now depends on speed, awareness, and timing. As the butterfly practices, its wing beats become smoother. It learns how to angle its wings to glide. It learns how to land without folding them awkwardly. The body that was rebuilt inside the chrysalis begins to move with confidence. The change from crawling to flying is one of the most dramatic shifts in the animal world. One form lived to eat and grow. The other lives to travel and reproduce. What began as a slow-moving larva is now part of the open air. And this change is not just beautiful, it is useful. The change from caterpillar to butterfly may seem almost magical. One body crawls and eats leaves, the other flies and drinks nectar. But this transformation did not appear by accident. It exists because, over millions of years, it worked. Complete metamorphosis, also called holometabolous development, follows 4 main stages: egg, larva, pupa, and adult. The caterpillar is the larval stage. The chrysalis is the pupal stage. The butterfly is the adult. Each stage has a different job. This separation of jobs is one of the main reasons complete metamorphosis is so successful. The larva is built for growth. It eats constantly, It stores energy. Its body is soft and flexible, perfect for squeezing along stems and hiding among leaves. It does not need wings. It does not need to search widely for mates. Its only goal is to gather resources and grow. The adult has a different goal. It is built for movement and reproduction. It must find mates. It must spread to new areas. In many species, it feeds on a completely different food source than the larva. Butterflies drink nectar. Caterpillars chew leaves. Because they eat different foods, they do not compete with each other. This is a key advantage. If the larva and the adult needed the same food, they would compete for it. Young and mature individuals would struggle over the same leaves or flowers. By separating life into distinct stages with different diets and habitats, insects reduce competition within their own species. They also reduce crowding. The caterpillar usually stays close to the plant where it hatched. The butterfly can travel far away. It can lay eggs on many plants across a wide area. This spreads out the next generation, lowering the chance that all the larvae will be in one place. Complete metamorphosis also allows each body plan to specialize. The caterpillar's body is simple but efficient for growth. The butterfly's body is complex and designed for flight. If an insect had to keep the same body throughout its life, it would need to compromise. It might not grow as quickly as a caterpillar. It might not fly as well as a butterfly. By splitting life into stages, evolution allows each stage to focus on one purpose. This system is not unique to butterflies. Many insect groups use complete metamorphosis. Beetles, flies, wasps, bees, ants, and moths all follow this pattern. Together, these insects make up a large majority of all insect species on Earth. Scientists estimate that about 80 to 90% of known insect species undergo complete metamorphosis. This means that the strategy of separate larval and adult forms has been extremely successful. It has allowed insects to adapt to many environments and food sources. Over time, this led to adaptive radiation. This means one common ancestral form gave rise to many different species, each suited to a specific niche. Some larvae live underground, some live in water, some burrow into wood. Adults may fly, swim, or crawl in completely different habitats. By dividing life into stages, insects unlocked more ways to survive. There is another advantage. The pupal stage provides a protected period for change. Instead of slowly reshaping their bodies while still active and exposed, insects transform inside a casing. The chrysalis shields the process. This reduces risk during one of the most complex changes in biology. Of course, metamorphosis also carries danger. A chrysalis cannot escape predators. A caterpillar must survive long enough to reach pupation. A butterfly must survive long enough to reproduce. But overall, the benefits have outweighed the risks across evolutionary time. When you look at a butterfly flying across a field, you are seeing the result of this long evolutionary experiment. The leaf-eating stage and the nectar-drinking stage are not separate creatures. They are two parts of one life cycle designed to avoid conflict and increase survival. Complete metamorphosis exists because it works, and it works so well that it has shaped the balance of life on this planet. When you see a butterfly drifting across a garden, it can feel rare, light, delicate, almost separate from the rest of the living world. but the process that created it is not rare at all. Complete metamorphosis is one of the most common life strategies on Earth. Scientists estimate that around 80 to 90% of all known insect species go through complete metamorphosis. That includes not just butterflies and moths, but beetles, flies, bees, wasps, ants, and many more. These groups are everywhere. In forests, in fields, in cities, in deserts, in freshwater streams. In fact, beetles alone make up more species than almost any other animal group on the planet. And beetles undergo the same four-stage process: egg, larva, pupa, adult. The larval form may look nothing like the adult. Yet inside the pupa, a similar transformation unfolds. Flies do it, wasps do it, bees do it. Across the planet, tiny bodies are breaking down and rebuilding every day. This strategy has helped insects become one of the most dominant forms of life on Earth. In terms of species numbers, insects far outnumber mammals, birds, and reptiles combined. In terms of biomass, the total weight of insects across the planet is enormous. Many ecosystems depend on them. Insects pollinate plants. They break down dead material. They serve as food for birds, reptiles, fish, and mammals. Their life cycles are woven into the structure of forests, grasslands, and wetlands. And behind much of this success is metamorphosis. By separating life into stages, insects can use resources more efficiently. Larvae may live underground, while adults fly above ground. Larvae may feed on roots while adults feed on nectar. This spreads ecological roles across time and space. Because the larval and adult stages look and behave so differently, they can occupy different niches within the same environment. One species can play multiple roles over its lifetime. Multiply that by millions of species. The result is a planet filled with transformation. If you imagine a single forest, you might picture birds in the trees and deer moving quietly below. But beneath the leaves, in the soil, inside rotting wood, and attached to stems, countless insect larvae are growing. Some will spin cocoons. Some will harden into chrysalises. Many will emerge as winged adults. This happens quietly, almost invisibly. A caterpillar hanging under a leaf is easy to overlook. A pupa, buried in soil, is almost impossible to see. Yet these hidden stages are happening all around us. The scale is difficult to picture. In a single meadow during summer, thousands of insects may be in different stages of metamorphosis at the same time. Some eggs are hatching, some larvae are feeding, some pupae are suspended in stillness, some adults are flying. It is a constant cycle. This cycle has shaped ecosystems for over 300 million years. Holometabolous insects, those that undergo complete metamorphosis, appeared long before humans. Their ability to separate growth from reproduction allowed them to adapt to changing climates and landscapes. As continents shifted and plants evolved, insects diversified alongside them. Flowering plants and pollinating insects influenced each other's development. As plants form nectar, insects evolve structures to drink it. As insects evolve flight, plants benefited from wider pollination. Transformation became a shared rhythm in nature. Yet despite its global importance, metamorphosis often feels personal. When we watch a butterfly emerge from a chrysalis, It seems like a small, private event, a single shell opening, a single body expanding its wings. In reality, it is part of a vast, ongoing pattern. Across forests in South America, across fields in North America, across rice paddies in Asia, across grasslands in Africa, insects are transforming right now. Billions of quiet changes unfolding inside sealed casings. And yet, for all their success, insects remain vulnerable. A chrysalis cannot move. A pupa buried in soil depends on stable conditions. A shift in temperature, a change in habitat, or the loss of host plants can interrupt the cycle. Metamorphosis has allowed insects to spread across the globe. It has made them one of the most diverse and numerous groups of animals alive today. But each transformation still depends on balance, on the right plant, on the right season, on the right moment, on a planet that allows change to unfold quietly, one hidden body at a time. The chrysalis hangs quietly beneath a leaf or against a branch. It does not move. It does not feed. It cannot escape. Stillness invites danger. During the larval stage, a caterpillar can at least crawl away from a threat. It can drop from a leaf or hide along a stem. During the adult stage, a butterfly can lift into the air, but inside the chrysalis, there is no movement. The body is sealed within a casing, committed to transformation. Predators know this. Birds may peck at exposed chrysalises, breaking open the casing to reach the soft body inside. Small mammals may chew through cocoons hidden in leaf litter. Even ants can attack if they discover a vulnerable pupa. Yet one of the most precise threats comes from parasitic wasps. Some wasps specialize in finding caterpillars or pupae. They lay their eggs inside or on the body of the host. When the wasp larvae hatch, they feed on the developing insect. In some cases, they avoid vital organs at first, keeping the host alive long enough to support their growth. Eventually, the host does not survive. This may sound harsh, but it is part of the balance within ecosystems. Parasitic wasps help control insect populations. Without predators and parasites, caterpillar numbers could grow too high, stripping plants of leaves faster than they can recover. The pupal stage carries a high risk of mortality. Many caterpillars never reach adulthood. Even after surviving weeks of feeding, wandering, and attachment, the stillness of the chrysalis can become their most vulnerable moment. And yet, metamorphosis continues. How does the species survive if so many individuals are lost? Through numbers. Butterflies and moths lay many eggs. A single female may lay dozens, hundreds, or even more depending on the species. Most will not survive to adulthood, but enough will complete the cycle to keep the population stable. This strategy is common in nature. High mortality, balanced by high reproduction, creates equilibrium. It allows ecosystems to absorb losses without collapse. There is also camouflage. Many chrysalises blend into their surroundings. Some resemble dried leaves. Others look like twigs or small bumps on bark. Their colors match the environment. Their shapes avoid drawing attention. A predator scanning branches may overlook them completely. Timing helps as well. Some species pupate in hidden places, such as under leaves, within curled foliage, or beneath soil. Others synchronize emergence so that many adults appear at once. When large numbers emerge together, predators cannot consume them all. This increases the chance that some will survive to reproduce. Even with these strategies, the risk remains real. Stillness is a trade-off. The chrysalis protects the complex internal rebuilding process, but it also limits escape. The transformation must be completed within that fixed space. And yet, the risk serves a purpose. Caterpillars feed on leaves. They transfer plant energy into stored nutrients. Adults drink nectar and in doing so, pollinate flowers. The life cycle connects different parts of the ecosystem. Predators and parasites play their roles too. Birds feed their chicks on caterpillars. Wasps regulate insect populations. Each stage of metamorphosis becomes food or support for something else. The risk of becoming is part of a larger exchange. If you picture a single chrysalis hanging quietly, it may seem fragile, but across an entire forest, thousands hang at once. Some will be eaten, some will host parasites, some will complete their transformation. The balance holds. Metamorphosis is not designed to guarantee survival for every individual. It is shaped to maintain continuity across generations. The caterpillar gathers energy from leaves. The adult returns energy to flowers through pollination. The risks in between are woven into the system. Stillness may invite danger, but it also allows change. And that change links leaf to flower, one life stage to the next. A caterpillar chews quietly on a leaf. A butterfly rests lightly on a flower. Two diets, one life. At first glance, the connection between these two stages may not seem obvious. The caterpillar eats leaves. The butterfly drinks nectar. One is a herbivore that can strip a plant of foliage. The other is a pollinator that helps plants reproduce. Yet both roles belong to the same organism at different points in its life. The caterpillar stage is built for feeding. Its strong mandibles slice through plant tissue. Its long digestive tract processes cellulose and stores energy. Leaves are rich in nutrients, especially when eaten in large amounts. Over weeks of steady chewing, the caterpillar gathers the fuel it will later need inside the chrysalis. This feeding is not random. Many caterpillars specialize in certain plants. Some can only survive on one type of host plant. The chemicals in those leaves shape their growth and even their defenses. A well-known example is the monarch butterfly and milkweed. Monarch caterpillars feed almost exclusively on milkweed plants. Milkweed contains chemicals that are harmful to many animals. The caterpillar can tolerate these chemicals and stores some of them in its body. When the monarch becomes an adult, those stored compounds help make it unappealing to predators. The bright orange and black wings serve as a warning. In this way, the leaf stage prepares the flower stage. When the butterfly emerges, its mouthparts are no longer suited for chewing leaves. Instead, it has a long proboscis designed to reach deep into flowers and drink nectar. Nectar is a liquid rich in sugars. It provides energy for flight and reproduction. As the butterfly moves from flower to flower, it brushes against pollen. Some of that pollen sticks to its body. When it visits the next flower, pollen may transfer. This helps plants reproduce. Seeds form, fruits grow, new plants spread. Energy moves through the ecosystem. The sun powers plant growth. The caterpillar eats the plant and stores that energy in its body. During metamorphosis, that stored energy rebuilds the insect into an adult. The adult drinks nectar and helps the plant produce seeds. Those seeds grow into new plants, which will feed future caterpillars. Leaf and flower are connected through a single life cycle. This connection also shapes evolution. Plants and insects have influenced each other for millions of years. Some plants produce leaves with specific chemical defenses. Certain caterpillars evolve ways to handle those chemicals. Flowers develop colors and shapes that attract particular pollinators. Insects evolve vision and feeding structures that match those flowers. It is a long history of adjustment and response. The monarch and milkweed relationship is only one example. Many butterflies lay their eggs only on plants that can support their caterpillars. The adult must find the right host plant, guided by smell and taste receptors on its feet. The choice determines whether the next generation will survive. At the same time, flowering plants benefit from pollinators. Butterflies are not as efficient as bees at pollination, but they still play an important role, especially in open, sunny environments. Their long proboscis allows them to reach nectar in deep flowers. As they feed, pollen transfer becomes part of the exchange. The larval stage and adult stage do not compete. They support different parts of the ecosystem. The caterpillar may reduce leaf mass on a plant, but rarely does it destroy an entire population. The plant grows back. The butterfly helps that plant spread its genes. The balance continues. If you look at a single butterfly landing on a flower, you are seeing the final step in a chain that began with a leaf. The energy gathered as a crawling larva now powers flight. The body rebuilt inside the chrysalis now helps carry pollen between blossoms. 2 diets, 1 life. The logic of temporary forms becomes clearer here. The caterpillar's body was designed for leaf eating. The butterfly's body was designed for nectar and movement. Each form plays a part in the larger system. Metamorphosis does not break the connection between stages. It links them. From leaf to chrysalis to flower, the cycle holds together. And through that cycle, plants and insects continue shaping each other, season after season. The caterpillar was built to eat. The butterfly was built to fly. Both bodies were temporary. When we look at metamorphosis, it can feel dramatic. One form disappears. Another takes its place. But beneath that visible change is a steady design. Each body exists for a specific purpose. Each is meant to be replaced. The larval body is a tool for growth. A caterpillar does not need wings. It does not need long-distance vision. It does not need to search for mates. Its body is soft, flexible, and efficient at turning leaves into stored energy. The digestive system is large. The muscles are arranged for crawling. The head is shaped for chewing. Everything about the caterpillar supports one goal: grow as much as possible, as quickly as possible. Once that goal is complete, that body is no longer the best design. The adult body is a tool for movement and reproduction. The butterfly's digestive system is narrow and built for liquid food. The mouth has become a coiled tube. The thorax holds strong flight muscles. The wings are broad and light, covered in scales that can reflect color and signal to mates. The eyes are large and made for detecting motion and color. The butterfly does not need to grow larger. It does not need to chew leaves. It needs to travel, find nectar, and lay eggs. 2 stages, 2 specializations. If one body had to perform both tasks, it would be less efficient. A creature that tried to grow rapidly and fly long distances at the same time would face trade-offs. Wings are expensive to build and maintain. Large digestive systems add weight. A compromised body would not perform either job as well. Metamorphosis solves that problem by dividing life into phases. First, build mass. Then, use it. This separation makes development more efficient. The caterpillar gathers resources without investing in adult structures too early. The imaginal discs wait quietly, while the larval body focuses on feeding. Only when enough energy is stored does the rebuilding begin. The genes guiding this process remain the same throughout. The caterpillar and butterfly share the same DNA. What changes is which genes are active at a given time. During the larval stage, growth-related genes are active. During pupation, reconstruction genes turn on. During adulthood, reproductive and flight-related genes dominate. The blueprint does not change. The instructions are timed. This timing allows each stage to be optimized. The caterpillar's body can be simple and expandable. The butterfly's body can be precise and structured. Neither form is permanent. Each is a step toward the next. It may feel as though one body dissolves and another appears from nothing. But the continuity is never broken. The nervous system remains connected. The imaginal discs were present from the beginning. The energy used to build wings was gathered while chewing leaves. Transformation is not an accident. It is strategy. Across millions of years, insects with complete metamorphosis thrived. Those that separated growth from reproduction gained flexibility. They could adapt to different habitats, climates, and food sources. They could spread widely. The temporary nature of each body becomes part of the advantage. The caterpillar is not meant to last. It is meant to prepare. The chrysalis is not meant to endure. It is meant to reshape. The butterfly is not meant to grow larger. It is meant to continue the cycle. Each form serves a lasting outcome. Survival of the next generation. When the butterfly lays eggs on a host plant, it begins the process again. Those eggs will hatch into caterpillars that do not resemble their parent at all, and yet they carry the same instructions. Temporary bodies, continuous lineage. If you step back, the pattern feels calm. There is no rush in the design. Growth unfolds. Dissolution follows. Reconstruction completes. Flight begins. Eggs are laid. The cycle turns. The caterpillar was always meant to become something else. The butterfly was always built from what came before. And in that steady replacement of one form by another, the logic of metamorphosis becomes clear. Not chaos, not magic, but design shaped by time. When we first hear that a caterpillar breaks down inside a chrysalis, it can sound chaotic. Muscles dissolve, organs reorganize. One body disappears, another takes shape. But inside that sealed casing, there is no chaos. There is order. Metamorphosis is guided by hormones that rise and fall in precise amounts. Two of the most important are juvenile hormone and ecdysone. During the larval stage, juvenile hormone keeps the body in growth mode. Each time the caterpillar molts, this hormone tells the body to remain a caterpillar. When juvenile hormone levels drop and ecdysone rises in a new pattern, the message changes. Growth stops. Transformation begins. These signals are not random bursts. They are timed carefully. Cells throughout the body respond to them in predictable ways. Some cells begin shutting down larval functions. Others prepare to divide and specialize into adult tissues. Cellular communication continues every second. Cells send chemical signals to nearby cells. Proteins bind to receptors. Genes are turned on or off. The imaginal discs expand in specific directions. Muscle cells attach at precise points. Veins form inside wings following established pathways. Even during the semi-fluid phase, structural integrity is maintained. The tracheal system continues delivering oxygen. The dorsal vessel keeps circulating hemolymph. The nervous system does not vanish. It adapts. Programmed cell death, or apoptosis, is part of this order. Cells that are no longer needed do not simply decay. They dismantle themselves in steps. Their components are packaged and recycled. Nothing spreads randomly. Nothing spills out of control. Timing matters at every level. If hormones rise too early, development can fail. If temperature shifts too suddenly, the process can slow or stop. Yet over millions of years, natural selection has refined this system. Those individuals with more reliable timing survived. Those with poorly regulated transitions did not. The result is a process that appears dramatic from the outside, but is steady and measured within. Inside the chrysalis, wings do not grow in tangled shapes. They form symmetrically. Left mirrors right. Patterns align. Pigments appear in organized bands. Flight muscles attach in balanced pairs. Even the coiled proboscis forms from two separate halves that fuse together with precision. The body never loses its overall structure. It changes it. Think of it as remodeling a house without tearing down the foundation. Some rooms are removed. Others are built. The wiring is adjusted. The plumbing is rerouted. But the core frame holds steady. Metamorphosis follows that kind of logic. The genetic instructions are present from the beginning. The egg contains the full blueprint for both larval and adult forms. The difference lies in which parts of that blueprint are active at a given time. During larval growth, genes for feeding and expansion dominate. During pupation, genes for reconstruction take control. During adulthood, genes for movement and reproduction guide behavior. Each phase flows into the next. There is no urgency in the chrysalis. Development proceeds at a pace shaped by temperature and season. Warmer days may speed up chemical reactions. Cooler nights may slow them. Yet the sequence remains the same. Order within change. Even the final emergence follows timing. The casing weakens along a seam. Muscles push outward in coordinated contractions. Hemolymph flows into wings along established channels. The wings dry and harden in sequence. Nothing is rushed. Nothing is accidental. When we watch a butterfly lift into the air, we are seeing the visible result of invisible coordination. Millions of cells acted in harmony. Signals rose and fell in careful balance. Structures dissolved and rebuilt without losing integrity. Evolution did not design this overnight. It refined it over vast stretches of time. Small improvements in timing and structure accumulated. Those refinements made transformation more reliable, more stable, more precise. What looks like a complete change of identity is, at its core, a continuous process guided by instruction. The caterpillar did not become chaos before becoming a butterfly. It followed a program shaped by millions of years of testing. Inside the chrysalis, change is steady, not wild, not uncontrolled, but guided. And in that guidance, there is a quiet kind of calm. A caterpillar begins as a small body with one clear purpose: eat and grow. It moves slowly across leaves, storing energy from sunlight captured by plants. It sheds its skin again and again, each time expanding a little more. Inside that simple body, the blueprint for something else is already present. Then comes stillness. The chrysalis forms. Movement stops. The outer world fades. Inside, tissues dissolve in careful steps. Cells communicate. Structures reorganize. What once crawled prepares for flight. There is no rush in this process. Hormones rise and fall. Temperature shapes the pace. Wings form in darkness. Muscles anchor. Patterns settle into place. When the shell finally splits, the butterfly does not burst into the air immediately. It hangs quietly. It expands. It dries. It waits until its body is ready. One form gave way to another, not by accident, not by chaos, but by sequence. The caterpillar was never meant to stay a caterpillar. The chrysalis was never meant to last. The butterfly will not remain forever either. Each stage serves its moment. Each prepares the way for the next. Across forests, gardens, and fields, this quiet logic unfolds every season. Leaves become fuel. Fuel becomes wings. Wings carry pollen. Eggs return to leaves. The cycle continues. Change, in this story, is not disruption. It is design shaped by time. And as you rest now, you might picture a single chrysalis hanging beneath a leaf. Still, patient, complete in its pause. Inside, something is becoming. There is no urgency in it, only sequence, only timing, only the quiet logic of becoming.